Paracoccus denitrificans introduction. What is Paracoccus denitrificans? Paracoccus denitrificans is a gram-negative bacterium capable of reducing nitrate to nitrogen gas via the denitrification cycle, operating in both aerobic and anaerobic conditions. This species has a genome of approximately 4.5 megabase pairs encoding the denitrification operon and respiratory enzyme systems. It is applied in wastewater treatment and used as a microbial model organism in research. General introduction to Paracoccus denitrificans. Paracoccus denitrificans is a gram-negative, non-spore-forming bacterium belonging to the Rhodobacteriaceae family. First identified in 1910 for its ability to convert nitrate to nitrogen gas, this species has become a quintessential model for studying the nitrogen cycle and bacterial respiratory mechanisms. Reference: Zunft, 1997. Its flexible metabolic capabilities allow Paracoccus denitrificans to live in both aerobic and anaerobic conditions, utilizing different electron acceptors for ATP synthesis. Its heterotrophic and autotrophic nature aids research into electron transfer mechanisms, oxidative phosphorylation, and low-light photophosphorylation. The scientific role of Paracoccus denitrificans is not limited to nitrate reduction, but extends to understanding the structure of the electron transport chain similar to mitochondria, protein modification mechanisms, and applications in nitrogen gas emission treatment. Morphological and physiological characteristics. Paracoccus denitrificans has a spherical or oval shape, with a diameter ranging from 0.6 to 1.0 micrometers. The cell surface has a typical gram-negative outer membrane containing lipopolysaccharides and porin proteins that facilitate metabolism. This species exhibits strong aerobic respiration when oxygen is available, using an electron transport chain to generate energy. When oxygen is depleted, it quickly switches to an anaerobic mechanism, using nitrate or nitrite as the terminal electron acceptor, maintaining oxidative phosphorylation at an efficient level. For aerobic life, the electron transport chain includes complexes 1 through 4, homologous to eukaryotic mitochondria. For anaerobic life, it uses the denitrification enzyme system, specifically the enzymes NAR, NER, NOR, and NOS, to reduce nitrate or nitrite. Additionally, it forms biofilms on the bottom surfaces of water systems or soil particles, contributing to cell protection and microbial community maintenance. Nutritional requirements and culture conditions. Paracoccus denitrificans grows optimally at neutral pH between 6.5 and 7.5 and a temperature of 28 to 30 degrees Celsius. Under standard conditions, LB medium or MSS1 medium supplemented with nitrate is used to stimulate both aerobic and anaerobic pathways. Let's look at the optimal growth conditions. For pH, the ideal range is between 6.5 and 7.5. This is typically adjusted using HEPES or a phosphate buffer. For temperature, the culture should be kept between 28 and 30 degrees Celsius, which is most stable in a humidified incubator. Regarding its carbon source, it can utilize acetate, lactate, or ethanol at a concentration of 0.5 to 1% weight per volume. Finally, to activate its denitrification capability, you need a nitrate source, specifically sodium nitrate, at a concentration of 10 millimolar. Additionally, Paracoccus denitrificans can utilize glucose or succinate as carbon sources in oxygen-rich environments. In anaerobic cultures, the addition of molybdenum and iron salts aids in the synthesis of denitrification system enzymes. Regarding anaerobic electron acceptors, it uses nitrate and nitrite. Trace elements required include molybdenum, iron, and copper for enzyme active center constitution. Vitamin and amine supplementation, such as biotin and thiamine, are occasionally required for genetic studies. Mechanism of nitrate reduction and denitrification. Denitrification is a chain of reduction reactions converting nitrate into molecular nitrogen, passing through intermediate stages including nitrite, nitric oxide, and nitrous oxide. Each step is catalyzed by a specific enzyme. The first step is reduction of nitrate to nitrite. In this reaction, nitrate plus 2 protons plus 2 electrons yields, via the NAR enzyme, nitrite plus water. The second step is reduction of nitrite to nitric oxide, catalyzed by nitrite reductase, also called NER. The third step is reduction of nitric oxide to nitrous oxide via nitric oxide reductase, also called NOR. The fourth and final step is reduction of nitrous oxide to nitrogen gas, catalyzed by nitrous oxide reductase, also called NOS. Electron flow through each enzyme is linked to the proton transport system, creating a transmembrane proton gradient and synthesizing ATP via ATP synthase. The energy efficiency of this anaerobic process is equivalent to 60 to 70% of aerobic respiration. Metabolic pathways and respiration, Paracoccus denitrificans operates a flexible respiratory system, allowing the use of oxygen or nitrate intermediates in oxidative phosphorylation. Under aerobic conditions, electrons are transferred through an electron transport chain consisting of complex 1, which is NADH dehydrogenase, complex 2, which is succinate dehydrogenase, complex 3, which is cytochrome BC1, and complex 4, which is cytochrome C oxidase, ultimately reducing oxygen to water. Under anaerobic conditions, Paracoccus denitrificans switches to the denitrification cycle. This flexibility provides an ecological advantage, allowing growth in diverse environments and oxygen-depleted conditions. To compare the energy efficiency of these pathways, aerobic respiration using oxygen yields an energy potential of approximately 3 ATP per 2 electrons. In contrast, denitrification using nitrate yields about 2.1 ATP. Using nitrite is slightly less efficient, yielding approximately 1.8 ATP. The electron transport chain has a diverse organization, and some enzymes resemble those found in eukaryotic mitochondria. A proton gradient is built across the cell membrane, powering ATP synthase. Regulation occurs through feedback based on oxygen and nitrate levels, which controls the expression of respiratory enzyme genes. Genetic structure and genome. The genome of Paracoccus denitrificans strain PD1222 is approximately 4.5 megabase pairs in size, consisting of one main chromosome and 3 plasmids ranging from 150 to 200 kilobase pairs. The genome encodes over 4,300 open reading frames, many related to denitrification including the NAR, NER, NOR, and NOS operons, as well as electron transport systems. The denitrification operons are organized into modules. The NAR-GIJ module handles nitrate reductase. The NER-S module handles nitrite reductase. The NOR-CB module handles nitric oxide reductase. And the NOS-ZD module handles nitrous oxide reductase. Upstream regulatory regions contain FNR-like and DNR elements responsive to oxygen and nitric oxide levels. The genetic material is divided across 4 main parts. First, the main chromosome is 4.2 megabase pairs and contains the core genes for metabolism and respiration. Second, plasmid P-PD 1222-1 is 190 kilobase pairs and contains regulatory genes for denitrification. Third, plasmid P-PD 1222-2 is 160 kilobase pairs and manages stress response and chemical modification. Finally, plasmid P-PD 1222-3 is 150 kilobase pairs and handles self-replication elements and antibiotic resistance. Reference genome data is accessible at NCBI accession GCF_0000091145.1 and Uniprot proteome up 000000585. Environmental role and the nitrogen cycle Paracoccus denitrificans plays a crucial role in the global nitrogen cycle, converting excess nitrate in soil and water into nitrogen gas, thereby reducing nitrate pollution and the greenhouse gas nitrous oxide. Denitrification activity helps balance nitrogen content in the biosphere and limits eutrophication, which is the over-enrichment of water bodies with nutrients. In natural ecosystems, Paracoccus denitrificans often resides in moist soil layers and pond sediments where oxygen levels fluctuate. Biofilm formation supports the creation of ideal microenvironments for continuous nitrate reduction. For wastewater treatment applications, combined aerobic and anaerobic tanks are used to optimize nitrate removal. The bacterium's highly efficient nitrous oxide reductase enzyme limits greenhouse gas emissions. Overall, it converts nitrate to inert gas, preventing pollution in rivers and lakes and contributing significantly to environmental balance. Biological and industrial applications, thanks to its heterotrophic nature and respiratory regulation, Paracoccus denitrificans is applied in biotechnology for producing nitrogen metabolizing enzymes, pharmaceutical precursors, and biofuel. Cell-free extracts from this species provide a favorable environment for producing proteins with complex post-translational modifications. Recent research focuses on genetic engineering to increase nitrate reduction efficiency, optimize electron flow, and produce green energy. CRISPR-Cas9 technology has been applied to edit the NAR and NOS operons, improving nitrous oxide reduction capacity by up to 40%. The reference DOI is 10.1038/S41598-019-45609-1. Applications include cell-free culture systems using modified PUR Express to increase post-translational modification rates. In molecular biology, the plasmid vector PPD1222-1 serves as a backbone for recombinant protein expression. Bioelectrochemical systems use microbial electrochemistry for nitrate reduction in industrial waste streams. Research and analytical methods *research on Paracoccus denitrificans combines genetic analysis, proteomics, and imaging techniques. 16S ribosomal RNA sequencing and multi-locus sequence typing allows strain identification, while ribosome profiling and liquid chromatography tandem mass spectrometry provide quantitative data on gene expression and the proteome. CRISPR interference, also called CRISPR, is applied to regulate the expression of denitrification operons, combined with quantitative PCR for messenger RNA measurement and enzyme assays to gauge nitrate reductase and nitrous oxide reductase activity. As for the specific research methods employed, 16S ribosomal RNA sequencing is used for strain identification, primarily with the Illumina MiSeq tool. Ribosome profiling provides codon-level resolution using deep sequencing. Proteome profiling relies on LC-MS-MS and an Orbitrap mass spectrometer. And finally, CRISPR is used for gene expression regulation utilizing the dCas9 and sgRNA system. Research potential and future directions ** future research focuses on integrating single-cell omics technologies to observe heterogeneity within Paracoccus denitrificans communities, particularly in biofilms. Microfluidics combined with fluorescence microscopy will enable real-time monitoring of denitrification kinetics. Mathematical modeling and deep learning algorithms will be used to simulate gene regulatory networks and predict responses to environmental changes. The combination of transcriptomics, proteomics, and metabolomics data promises to optimize cell-free systems for producing nitrogen metabolizing enzymes. In the field of renewable energy, researchers are developing bioelectrochemical reactors using Paracoccus denitrificans to combine nitrate reduction with electricity generation. This approach simultaneously supports wastewater treatment and green energy production, representing a promising direction for sustainable technology development.